that, I will move on to introduce our trained speaker, Heta Patel. Heta started her academic journey in University of California, Santa Cruz, after getting her studies in BS Bachelor's and Molecular Biology there. She moved to Netherlands, to Rotterdam, to get her PhD. She's working in a Netherlands Cancer Institute, NKI, in Amsterdam, Netherlands. And she's working with Tineke Lenstra, and her work is on the studying the influence of DNA supercoiling and transcriptional bursting using the single molecule imaging. And we are really delighted to have you here, Heta. Floor is yours. And you can ask questions as soon as Heta finishes, either by raising your hand or writing Q&A box or in the chat. Floor is yours, Heta, sorry.
Yeah, okay. Thanks, Liz, for the introduction. And also thanks to the organizers for this opportunity. So today I want to talk about how DNA supercoiling and topoisomerases can influence eukaryotic gene expression. So when most of us think of DNA, we think of this iconic double helix shape, but in living cells from bacteria to humans, DNA rarely exists as a double helix because it's continuously contorted for various nuclear processes. And one such process is transcription, where an elongating polymerase unwinds the DNA so it can copy the DNA to RNA. And as the polymerase elongates, it overwinds the DNA ahead and underwinds the DNA behind. And this deviation of the DNA structure from its relaxed state is known as DNA supercoiling. So transcription generates DNA supercoils, but DNA supercoils can also differentially influence transcription. For example, underwound DNA can promote initiation by making the DNA more accessible to regulatory proteins. And overwound DNA fosters elongation by destabilizing proteins so the polymerases can progress. However, cells must maintain optimal levels of DNA supercoils because excess supercoils can actually inhibit transcription. And what's really interesting is that in bacteria, the accumulation and release of DNA supercoils can stochastically switch genes on and off, resulting in a phenomenon known as transcriptional bursting. So bursting has been observed from bacteria to humans, and it's a fundamental property of genes where transcription occurs in pulses during an on period, and these on periods are interspersed by off periods where there is no transcriptional activity. So DNA supercoiling is the main driver of transcriptional bursting in bacteria, but whether bursting drives or even influences, whether supercoiling drives or even influences bursting in eukaryotes is unknown. So that's our main question. And so we want to know if supercoiling is driving bursting of neighboring eukaryotic genes, because it's been also shown that supercoils can travel on the DNA up to 1.5 kilobases. So to study this question, we're focusing on the gal gene cluster in budding yeast, which composes of 3 genes that are all well studied and highly regulated by the same transcription factor, gal4. And within the locus, gal1 and gal10 are divergently oriented, and gal10 and gal7 are tandemly oriented. And this locus is an ideal model system because all 3 genes are highly expressed, which means that there is high amounts of supercoils being produced at this locus. All 3 genes exhibit bursting, and the intergenic distances are 700 base pairs, which is within the 1.5 kilobases that supercoils have been reported to propagate. So to study the transcription dynamics of these genes, we utilize the MS2PP7 system, which is an orthogonal RNA labeling system that allows us to visualize the transcription of 2 genes at a time. And the way it works is that these DNA motifs are inserted at the endogenous genes of interest. And when these repeats are transcribed, the RNAs form these secondary stem loops, which are then bound by a fluorescent coprotein. And using fluorescence microscopy, this allows us to visualize single molecules of RNAs within living cells. And we're mainly interested in this brighter spot in the nucleus, which is the transcription site, which is the site on the DNA where nascent RNAs are actively being produced. So to study the temporal relationship between 2 neighboring genes, we perform dual color imaging on these yeast cells, and we track and quantify the fluorescence intensities of these transcription sites. And in this example trace, you can see that sometimes these 2 genes initiate together, whereas other times they don't. So to objectively quantify how similar these two signals are over time, we compute cross-correlation functions. And these are computed by shifting one signal with respect to another by a specific time lag. And so in this example, one of the signals can be shifted to the right to obtain one side of the function, but the signal A can also be shifted to the left. And this gives us a full cross-correlation function. So peaks represent positive correlations and valleys represent negative correlations. The amplitude tells us the degree of the similarity between the two signals, and the time delay tells us when the similarity occurs. So when we compute the cross-correlation functions for the divergent genes, we observe a peak at time delay 0, and this indicates that these two genes transcribe or initiate simultaneously. And this correlation decays to 0 around -100 and +100 seconds. And we know that this is the time it takes for these two genes to transcribe. So these divergent gal genes, when they're next to each other, initiate and transcribe simultaneously. And as a control, we put these loops onto different alleles where the expression of these two genes should be independent and therefore uncorrelated. And as expected, we get a flat line that indicates that there's no temporal correlation. So we use the same approach to also look at the tandem gene pairs. And there we observe a weak positive correlation that's not really present in the random control. And this indicates that despite having separate promoters, these tandem genes can also initiate simultaneously. So in wild-type conditions, the initiation of these divergent and tandem gene pairs is temporally coupled more than expected by random chance. And we wanted to know if DNA supercoiling played a role in this coupling. So to test this, we wanted to perturb DNA supercoiling levels and check how that affects this correlated initiation. And to perturb DNA supercoiling levels, we conditionally degraded topoisomerases, which are enzymes that relieve DNA supercoils. So yeast have two main topoisomerases, top1 and top2, that are involved in transcription and have the dual role of relieving both underwound as well as overwound DNA. And to deplete these proteins, we tag them with a degron, such that in the presence of a plant hormone auxin, these proteins are targeted for degradation. And when we quantified the top1 and top2 levels in wild type conditions, as well as in the absence and presence of auxin, we observed that in the absence of auxin, there's basal degradation of top1 and top2. And the presence of auxin, both the proteins are effectively degraded. So we exploited this basal degradation to get a range of yeast strains with varying levels of topoisomerases and therefore varying levels of supercoiling accumulation. And when we perform live cell imaging on these cells, we find that 50% of the cells are inactive for the expression of both these genes. But in the 50% that do show expression of the two genes, we computed the cross-correlation function. And compared to wild type, we still observe a peak at time delay 0, which indicates that these two divergent genes can still initiate simultaneously. But what's super cool is that we also start to observe these valleys at -100 seconds and +100 seconds. And so if you recall, 100 seconds is how long it took to transcribe these genes. And so these valleys indicate that the expression of one gene can inhibit the expression of the neighboring gene. So in other words, these two genes can sometimes transcribe simultaneously, but other times only one or the other gene is expressed because they're inhibiting each other. And we use the same approach for the tandem genes, of course, and there we also see that the tandem genes can initiate simultaneously, but upon topoisomerase depletion, only one or the other can initiate. So we wanted to know what results in this loss of correlated initiation of these neighboring gene pairs. So we looked at the relative transcriptional activity, or in other words, the expression levels of the three genes. And we observed that in partially depleted conditions, the middle gene GAL10 is significantly more inhibited than the flanking genes GAL1 and GAL7. And in fully depleted conditions, the transcriptional activity of all three genes is decreased, with GAL10 still being the most inhibited. And this is quite interesting because GAL1 and GAL10 share a promoter, but GAL10 is more inhibited than GAL1, And we reasoned that it's maybe because there's a highly expressed gene GAL7 downstream of GAL10, and hypothesized that supercoils from the transcription of GAL7 may be inhibiting GAL10 and therefore inhibiting the correlation of GAL1 and GAL10. And so to study the influence of this, we used a technique called single molecule FISH, which is where we fix the cells and hybridize them with fluorescent oligonucleotides that bind to the RNAs. And we're still interested in the transcription sites, or more specifically, the number of nascent RNAs at the transcription sites. And in this scatter plot, we show the GAL10 expression on the x-axis and the GAL1 expression on the y-axis, where each of these data points represents a cell. As a measure of correlated transcription, we compute the Pearson correlation in all the active cells in this scatter plot. And here's an aggregate of the correlations. It's just a dot plot where each data point represents a replicate. So we also confirmed with single molecule FISH that decreasing topoisomerase levels decreases the correlated initiation of the divergent genes. And we wanted to know, again, if the expression of GAL7 is inhibiting GAL10. So to test this, we scrambled the transcription factor binding sites in the GAL7 promoter and then measured the correlation of the divergent genes. And we observed that in wild type conditions, eliminating GAL7 expression does not affect the correlation of the divergent genes. But in topoisomerase deficient conditions, eliminating GAL7 expression actually rescues the correlation to wild type levels. So this indicates that in wild type conditions, yeast cells probably have sufficient levels of topoisomerases to minimize this inhibition. Whereas in topoisomerase deficient conditions, transcription generated supercoils from GAL7 are possibly inhibiting GAL10. And so to put this a bit more visually, we have underwound and overwound DNA in the promoter regions in the gene bodies. And based on the orientation, we reasoned that maybe it's underwound DNA that's inhibiting GAL10. So we wanted to check if the divergent genes are more inhibited by underwound DNA or overwound DNA. And to do this, we topically express bacterial topoisomerases, which can selectively relieve either underwound or overwound DNA in wild type and topoisomerase deficient conditions. So when we overexpress gyrase, which selectively relieves overwound DNA, we see a modest increase in the correlation of the divergent genes. But when we overexpress top A, which selectively relieves underwound DNA, we see a significant increase in the correlation of these two genes. And this indicates that the transcription of GAL1 and GAL10 is predominantly inhibited more by underwound DNA rather than overwound DNA. So far what I've shown you is just a 3-gene analysis, and we wanted to determine the genome-wide extent of the supercoiling-mediated inhibition. So we collaborated with Domenico Libri, whose lab has performed pull-to-crack-seq on these topoisomerase mutants. So crack-seq essentially gives us a genome-wide sequencing-based analysis of nascent transcription. So when we look at isolated genes, which are genes that don't have a neighbor within 500 base pairs, we see that supercoiling accumulation has no effect on the nascent transcription. So this is our control group. And now when we start looking at gene pairs, so genes within divergent gene pairs, as well as the upstream gene within tandem gene pairs, where negative supercoils are, underwound DNA is likely to play a role, we see a significant inhibition in nascent transcription for both of these groups. And when we look at the downstream gene of tandem gene pairs and the convergent gene pairs, we also see a substantial decrease in the nascent transcription. And here the overwound DNA is more likely to play a role. So these results indicate that both underwound and overwound DNA have inhibitory roles and suggest supercoiling mediated regulation of eukaryotic gene expression. So in summary, what I've shown you is that the transcriptional bursting of the gal genes is temporally coupled, and that this coupling actually requires a rapid release of DNA supercoils by topoisomerases. Additionally, the accumulation of DNA supercoils results in the mutual inhibition of adjacent genes, and I can hypothesize that it's not only transcription that results in this inhibition, but also maybe other nuclear processes such as replication or recombination or DNA repair, which also produce DNA supercoils and can therefore possibly inhibit nearby genes. I've also shown you that this supercoiling mediated inhibition is present genome-wide. And finally, unlike in bacteria, wild-type yeast maintains sufficient levels of topoisomerases to minimize this supercoiling mediated inhibition. And with that, I'd like to thank Tinika for the supervision and guidance, as well as the rest of my lab and the Division of Gene Regulation, as well as Domenico and Umberto for the CRAC-seq data and analysis. And thank you so much for your attention. I'm happy to take questions.
Okay, thank you, Heta, for this nice talk. Alison Borman has a question, so I will allow her to talk. You can unmute yourself and talk.
Thank you. Sorry, my internet's not the best, but hopefully you can hear me. Yeah, that was so cool. I love this idea of like supercoiling, regulating transcription. I just had a couple questions. So with the underwound DNA, like how does that relate to like negative supercoiling? Like, yeah,
underwound DNA is negative supercoils. I think I didn't want to confuse people with negative and positive, so I was sticking to underwound and overwound for negative and positive.
Gotcha. Yeah, so my other question was, do you think like some of these coils might also drive transcriptional bursting in some ways, like sometimes?
Yeah, I think if they're present in low levels, I think so there have been studies that have shown that negative supercoils enhance transcription. And I think at low levels, they can enhance transcription. But I think in our case, it's possible that there's hyper underwound DNA, and therefore it's inhibiting transcription.
Cool, thank you.
Yeah.
Thanks, Alison, for the question. So anybody else? I think Ben or Vlad also. Jordan has a question. Okay, go, Jordan.
Not very nice talk. Very interesting. What do you think is the?
Jordan, you cannot mute yourself.
Sorry, can you hear me now?
Yeah.
It was a very nice talk. What do you think is the mechanism behind this inhibition? Do you think it has to do with recruitment of transcription factors or more physical type of inhibition through restriction?
We've looked into multiple things so far. Short answer is it's still in progress, but we've looked at nucleosome stability and position, and we see that that doesn't change. We've also looked at GAL4 occupancy and the transcription factor occupancy and TBP occupancy, and that also doesn't seem to change. So right now, our reasoning is that it's something downstream of TBP binding. So currently, I'm performing single particle tracking on multiple PIC subunits to see if maybe one of those things changes. But basically, supercoiling is involved in every step of transcription. So I'm just trying to kind of tease out where this inhibition, the mechanism of this inhibition.
Okay, great, thank you.
Thanks, Jordan. So there's two questions in Q&A. One of them is from Edward Groh, asking, could you comment on tandem gene pairs and whether there is evolution at some genes to acquire sequence features that neutralize supercoiling interference between gene pairs? Heta?
Yeah.
Can you hear me?
Yeah, I can hear you.
Okay.
Yeah, I think so. So our observation mainly is that these tandem gene pairs are mutually inhibitory. And if there's some evolutionary advantage I'm not really sure. It's a good question. Maybe I can get back to you.
Okay, so then Sandipan also had a question. The Sandipan's nice talk. Where actually what the sorry, that really arty I didn't get it. So the differential effect you showed for GAL1 and GAL10, do you think that DNA sequence at the start of the genes may have any effect?
So the differential effects on GAL1 and GAL10. Oh, upon topoisomerase depletion. No, they share a promoter. So a lot of the elements are shared, except that they form their own picks. And so also with this perturbation, when we do the GAL7, when we mutate the GAL7 binding sites and we look at the expression of 1 and 10, we also see that not only does the correlation increase, but the expression of both the genes also increases. So I don't think it's really sequence dependent, or I don't really think how it could be, just because they do share the whole promoter.
Okay, thanks, Heta, and thanks everybody for questions. I think I will give the floor to Sky, so she would introduce our next speaker.